এখন ক্লিয়ার না?
জি স্যার।
Okay, let us start our today's makeup class, advanced makeup class on speed breeding for climate smart crop variety development. In our earlier class, we discussed on transgenic plants. How can we develop GM crop variety, as well as the application of frontier technology, genome editing. As the demand for food is increasing day by day due to several reasons. In case of Bangladesh, land decreasing, population increasing, and climate change posing a serious threat. So we need to accelerate our breeding process to develop climate smart crop variety to address the new demand of food security. Today, I shall discuss on what is speed breeding and what are the advantages and how can we speed up or accelerate our breeding uh, practices for getting crop variety resilient to climate change. So one of the important reason, target in front of us is sustainable development goal. And among the sustainable development goals, the goal two is to achieve zero hunger worldwide. And to achieve the zero hunger by the year 2030, really challenging. In that case, classical breeding and other breeding practices took very long time. So we need to speed up our breeding practices. And I shall also briefly talk about the breakthroughs in plant breeding. Already I discussed on the historical uh, development of plant breeding and precision breeding, especially genome editing. And strategies of speed breeding, especially I shall focus how we can combine uh, several breeding approaches for acceleration of the breed. Uh, I previously discussed this topic that climate change poses increasing risk to crop production through droughts, salinity, uh, pests, floods, and many other ways. And from 1981 to 2000, there was a comprehensive study on rice, maize, and wheat. It had been, found that over the year that long period, uh, crop yield increased 22 to 46% in Asia, Latin America, uh, and this is why we are feeding large number of population. But to meet the growing demand by 2050, when global population would be around 10 billion, a global increase in crop production of up about 100 to 110% is required. And to satisfy this demand, improved crop varieties are needed. So improved crops are urgently needed to meet growing demand for food and address changing climate change. Especially crop production is linked to several sustainable development goals, not only freedom from hunger, even end of poverty. If you see the farmers, growers, among the growers, poverty is a big problem, and it is also creating a pressure for, the productivity. Other thing is that smart agricultural practices is needed to have sustainable crop production, that is sustainable consumption and production also linked to the food security and sustainable ecosystem because modern agricultural practices are mostly based on uh, huge chemical and other inputs. So how can we reduce the inputs? Because some of those inputs like chemicals, agrochemicals like fertilizers and pesticides, they are creating serious pressure to the environment, causing environmental pollution. Now let us embrace the new technology, speed breeding, and it is an innovation. Uh, Dr. Lee Hickey, a professor at the University of Queensland, he actually uh, coined this term and developed this method, and it was published in Nature Plant Biotechnology Journal. Nature Biotechnology Journal. Stimulated by NASA aiming to grow wheat in space. Actually, NASA, they had a project to grow wheat in space. So it was a stimulating thinking because in the space, environment is quite different. Uses constant light or extended photoperiods and control temperature to accelerate plant development. Naturally daytime and nighttime. So naturally plant requires 16 hours daytime light period and 8 hours dark period for flowering. But if you change the this ratio, period of light period, then it has been found that you can accelerate the flowering of the plant. That means you can improve the breeding cycle, which is very crucial for reducing the time for breeding. So, what is speed breeding? Speed breeding technology that saves breeding time and resources through very rapid generation advancement in a control environment. Speed breeding obviously practiced like tissue culture in control environment. And in the conventional breeding, here you can see, and speed breeding, the image in the bottom. So in conventional breeding, a variety release takes 10 to 15, 15, 13, even 20 years. But in case of speed breeding, we can reduce it significantly, especially you see initial line development here takes 10 years. That means breeding line, and then you can do the field testing and variety release. So clearly here, 6 to 8 years you can reduce. How can you do it? By changing the light exposure to the plant.
স্যার
Uh, yes.
স্যার এখানে লাইন ডেভেলপমেন্ট বলতে কি বোঝায় স্যার?
Line development, for example, if you do the crossing or if you do the editing or if you do the genetic transformation, you will get the plant, and it takes few generation to get a candidate line. Line means from one plant you will get lots of seed, and those seeds are in a single line. So they are, when they are nearly homozygous, then it is called line ready for field testing. So line development, line is the candidate for future variety. And it is not guaranteed that all lines will be variety because it needs to be tested in the field. And also there are some regional field trials to confirm that the line you are claiming superior to the existing varieties are stable and in variable environments. So results in 3 to 9 generations per year compared to 1 to 2 generations per year. That means by speed breeding using the physico-chemical and photoperiod changing, you can reduce from 3 to 9 generation, generate 3 to 9 generations per year in the naturally actually 1 to 2 generation. For example, in case of wheat, we can only cultivate wheat in the winter. So every year one cycle only. But in control condition, through speed breeding process, you can generate up to 9 generations. So you can reduce 9 years job, you can do 1 year. Rapidly develop genetically pure and stable plants to accelerate release of new cultivars, new variety. So as several generation, you can get the stable breeding line. So why speed breeding is necessary? The world's population, as I mentioned earlier, is expected to increase at 20% over the next 30 years from 7.7 billion to 9.7 billion by 2050. And by then, we will need to produce 974 more calories per person per day. It's really challenging task. Increased water use and elevated temperature caused by climate change will negatively affect food production. So speed breeding can help to increase food production by increasing genetic gain. That means we do the breeding to get the genetic gain over time and producing crops that are acclimated to new environmental conditions more quickly. So in that case, we use speed breeding, even double haploid production, I told you earlier, and genomic selection and modeling to optimize the breeding pipeline. And also we can add, artificial intelligence and imaging technology, unmanned vehicle to uh, uh, get the data, not manually, but, mechanically. So major challenges in our country for food security, that adding 2 million people per year, I discussed in earlier lecture, to 23 to 25 crore people by the year 2050. Our land is very limited, 8.774 million hectares cultivable land, and loss of land 1% per year. This is really the pressing issues. And other additional challenges, biotic and abiotic stresses, salinity intrusion in the coastal belt, chronic and seasonal drought, flooding, limited organic matter in the soil, soil health is badly deteriorated, and post-harvest food loss. After harvesting, we have no processing and value addition practices. So these are the major challenges we have to address. But speed breeding cannot solve all the things. In crossing, conventional breeding, 3 to 7 years needs crossing and back crossing. And then testing in the field, 4 to 5 years and 1 to 3 years for seed increase and release to the farmers. So 15 years or more. But conventional cycle time of breeding is really slow. Here, how people do: parent one, parent two, cross F1, and then F2, lots of line, and they segregate, and finally F6, you can get the line, and then F6 line. What you do? You do the trial, and there are several things associated with variety development, because the trait you integrate, and you have to check really stable or profitable compared to the existing varieties, and also other things. New variety should have, other acceptance in the market, and also other, industrial acceptance. Traditional breeding, actually, really, Dr. Norman Borlaug, he did the traditional breeding. F6 up to F6 generation crossing is necessary to get the nearly homozygous line. But in case of speed breeding technologies, it shortened the breeding cycle. Um, how? Because you can get the homozygous line by using double haploid technology, Antharva or pollen culture. So then line will be pure, and you can go for the field testing. Another thing is genomic selection. If you do the genomic, for example, marker assisted breeding, or genome based, sequence based selection, then it can save your time. And obviously, computational modeling enables optimal design and breeding program. Nowadays, big number of plant population, if you want to take manually all data, phenotypic data is difficult. So we can use the computational method, even an unmanned vehicle, and you can do even modeling using artificial intelligence. And one of the integration of the speed breeding, CRISPR-Cas genome editing, which is much more precise than previous all methods. So these proven technologies, that means speed breeding is a combination of several proven uh, concepts and technologies, increase genetic gain per cycle and decrease time to release a new variety. So here you can see conventional took 10 to 12 years, but in accelerated breeding, 5 to 6 years. And it also save time as well as the cost because every trial requires huge energy, huge cost. So, rapid trait tracking through speed breeding and marker assisted selection. For example, you integrate a gene. So, you can check the gene whether in the progeny, not only morphology, and do the back crossing. So, marker assisted breeding, speed breeding here you see is taking a very short time. This is the field. Conventional breeding, even regular greenhouse also are not enough to speed up, but obviously it speed up some of the processes. And by using marker-assisted breeding, lots of genes have been integrated into the plant and develop the new variety, not phenotypically. What you do, extract the DNA in the progeny and check in the PCR which progenies carries the gene. And double haploid I already discussed, because when you get the F1, then you can make them homozygous by anther culture or pollen culture to get the haploid plant, and then by colchicine treatment you can get the fertile double haploid homozygous plant ready to field trial. Genomic selection, as I mentioned before, genomic selection in the parents, you can do GBS, genome-based sequencing. Lots of population, which one carries the gene, you can check them. So genomic selection optimizes the breeding values based on the incremental value of each genomic segment. Segments are identified by genetic variation that can be assayed using high throughput DNA sequencing technology, that is next generation DNA sequencing. And breeding values are estimated. That means the genetic code you can find out and then utilize for improving the getting the genetic gain for your target trait. There are lots of factors involved or should be considered when optimizing genetic gain per dollar invested. Rapid breeding cycle, high selection accuracy, selection of partners, parents with high breeding values. Breeding values means you target traits. Those contain your target traits, you pick up them. Model breeding programs to achieve optimal results are breeding cycle, number of crosses made, number of generations per year, number of candidates advanced per cross, and then testing up to 6 generation using double haploid and CRISPR, you can reduce the cycle. So supporting technologies for speed breeding, one is genotyping, another phenotyping. Phenotyping we can do by unmanned vehicle like drone, and obviously genomic selection. So modern crop improvement occupies, needs interdisciplinary approaches, cell biology, molecular biology, plant growth facilities, or plot vision by using automatic drone and automatic data analyzer. Obviously, omics and precision agriculture, high throughput sequencing help us. And in case of data management, different kind of simulation and modeling are used. So these are the combined and integrated approaches of several advanced technologies to accelerate the breeding. That is, accelerated breeding and speed breeding are the same term. So what are major advantages? Number one advantage is shortened breeding cycle. That is, breeding cycle time is reduced by decreasing time from new parental crossing to pure line development. Okay. And second thing is the breeding values incorporate performance of crops in current environment. So genomic selection considers performance of lines across multiple environments. So you can change the environment and you can accelerate the release of the variety. Third, new parental combinations based on complementary breeding values. And fourth is visual characterization time is reduced by discarding lines with low breeding values. So how can you discard them? Based on the imaging data you take by using drone. So those are not satisfactory traits in the segregating population. You can remove them. So and pick up the best lines that you desire. And finally, breeding pipeline is modeled to identify most efficient practices. So you can reduce the cost. You can reduce the cost of the, one second, please. So next is you may ask the question whether already been practiced or not. Yes, speed breeding has already been successfully applied in wheat, barley, canola, chickpea, brachi, many crops. So brachypodium, medicago. So if you are asked whether speed breeding being practiced in the real field, yes, it is not only the the how to say theory, it is in practice. And speed breeding sometimes use this type of before ripening, because ripening takes 7 or more days, 15 days. So what you can do before ripening, you can harvest, and by drying you can get this shriveled seed. A shriveled seed, no problem because they germinate, but you reduce the time of breeding. First wheat variety developed using speed breeding in Australia. And it is really amazing, and Dow AgroSciences, they did the job. Now it is becoming a popular technology worldwide. Speed breeding facilities now in many countries, even in Bangladesh, it is going to be introduced, and some of the laboratories are using. And even if you do the transgenic or CRISPR pipeline, you can combine the, especially light and other physicochemical parameters to accelerate the, flowering and reduce the breeding cycle. So opportunities, commercial breeding companies using speed breeding because they need profit, per dollar investment. Can we take speed breeding off the grid? It's a question because even in winter, severe winter and harsh environment, rainy season, you can do the job in control environment, and winter wheat still requires vernalization. Vernalization is, sometimes seed has the problem of flowering. So if you do cold treatment of the seed, especially winter wheat, then you can get flower. Otherwise, you cannot get the flower. Uh, Speed breeding, obviously, making a new paradigm shift in combination with the CRISPR and other smart technology. In India, they released genome edited variety, and they claim that they use the speed breeding technology. And speed breeding would be much more expedite once, a CRISPR method becomes the user friendly, like the Lihiki, he mentioned express edit approaches. Express edit approaches, that means no need tissue culture. You just spray CRISPR construct and develop the disease resistant variety or like that. So in future, Cas9 and guide RNA, you make the construct and then spray on the seedlings and edit the plant, and for example, disease resistance and pathogen spray. And if you see some plants are resistant and you can select it, that would be fantastic. So there are some short-term and long-term deliverables for speed breeding. Now, long-term, we can get the climate resilient crop variety, and our agriculture would be sustainable, food security would be sustainable, and obviously we will have trained people, new improved varieties. But short term, we can get the sequencing data and double haploid methods. These are we can target CRISPR genome editing, digital field imaging, because all these are requires for having the long term impact. So these are the short term goal we can set, and long term goal we can set for getting the genetic gain. So lots of challenging we have, because we need funding, as well as we need the international collaboration and experiences, train manpower, and then finally our soil is fertile, our environment is very good all year round. We can cultivate. Our farmers are also smart. Finally, we can have good uh, output from the speed breeding. So this is all about brief talk. Um, if you have any question, I can discuss, according to your question. So please ask some question.
স্যার ডাবল হ্যাপ্লয়েডের যে মেকানিজমটা ছিল অ্যান্থার কালচার পলেন কালচার, স্যার এই মেকানিজমটা বুঝি নাই।
এই মেকানিজমটা তো তোমার এর আগের ক্লাসে ডিটেইল আলাপ হয়েছে। এটা খুবই সিম্পল থিং। এই যে এখন দেখো এখানে আমরা অ্যান্থারকে যখন কালচার করি, এখানে অ্যান্থারকে যখন কালচার করি টিস্যু কালচারের মাধ্যমে, তখন আমরা বিভিন্ন হরমোন ট্রিটমেন্টের মাধ্যমে এরকম ক্যালাস তৈরি করতে পারি। তারপরে রিজেনারেশনের মাধ্যমে আমরা এরকম সিডলিং পেতে পারি। কিন্তু যেহেতু অ্যান্থার হচ্ছে শুধুমাত্র মেল সেক্স অর্গান, ফলে এখানে এন থাকবে, টু এন থাকবে না, হ্যাপ্লয়েড। এই হ্যাপ্লয়েড প্লান্ট হচ্ছে তোমার ইনফার্টাইল। দিস ইজ ওয়াই তাকে কলসিজিন দিয়া যদি ট্রিটমেন্ট করি, দেন ডাবল হ্যাপ্লয়েড হবে, এবং ডাবল হ্যাপ্লয়েড হচ্ছে ফার্টাইল। তো এখানে যেহেতু কোনো রকমের জেনেটিক রিকম্বিনেশন হচ্ছে না, পিওর প্যারেন্ট সেল থেকে এটা করা হয়েছে, ফলে এই অ্যান্থার কালচার তোমাকে ডাবল হ্যাপ্লয়েড হোমোজাইগাস প্লান্ট দিবে। এই হেটারোজাইগাস মানে রিকম্বিনেশন হয়ে, মনে করো যে যেটা সেগ্রিগেশন হওয়ার রিস্ক থাকে, এটা থাকবে না। অথবা তুমি পোল্যান্ড থেকে, শুধুমাত্র পোল্যান্ড, প্যারেন্ট পোল্যান্ড থেকে তুমি কালচার করে একই রকম ডাবল হ্যাপ্লয়েড পেতে পারো। তা আমরা যদি কোনো একটা ব্রিডিং করি, মনে করো ডিসিমিলার প্যারেন্ট, একটাতে ডিজিজ রেজিস্ট্যান্ট জিন আছে, আরেকটাতে নেই। এখন যেটাতে নেই এটা খুব হাই ইল্ডিং, ক্রসিং করার পরে F1 হবে। তো এই F1 মেন্ডেলিয়ান লতে 6 জেনারেশন পর্যন্ত তুমি যেতে হবে হোমোজাইগাস পেতে হলে। কিন্তু অ্যান্থার কালচার বা পোলেন কালচারে আমরা ভেরি কুইকলি হোমোজাইগাস ডাবল হ্যাপ্লয়েড ফার্টাইল প্ল্যান্ট পেতে পারি, এবং তুমি যে জিনকে ডিজায়ার করছো এই জিনও ক্যারি করতে পারে, স্ক্রিনিং করে বের করবা। এটা কি ক্লিয়ার হয়েছে তোমার কাছে?
জি স্যার।
এক্সেলেন্ট। আর কারো? আর কোনো প্রশ্ন আছে কিনা? আমি লক্ষ্য করেছি 24 জয়েন করেছে, ভেরি গুড। আছে কারো প্রশ্ন? যদি কোনো প্রশ্ন না থাকে, থ্যাংক ইউ সো মাচ। আমি ফাইল অলরেডি শেয়ার করেছি, এই ভিডিও রেকর্ডটাও তোমাদের সাথে শেয়ার করব, এবং তোমরা সবাই ভালো থেকো। আমি আজকে চায়না যাচ্ছি, 23 তারিখ ব্যাক করব আশা করি। এরপরে তোমাদের সাথে পরীক্ষার হলে 26 তারিখ দেখা হবে। হুম।
স্যার আমাদের
ততক্ষণ,
মিড টার্মটা স্যার 6:30 থেকে শুরু হবে না?
ইয়েস,
আচ্ছা
6:30 থেকে শুরু হবে, এবং তোমাদের যে প্রোটোপ্লাস্ট এন্ড প্রোটোপ্লাস্ট ফিউশন পর্যন্ত যে লেকচার হয়েছে, আমি ওই দিন ক্লাসের মধ্যে রিভিউ করেছি কি ধরনের প্রশ্ন হবে। কাজে ওইভাবে তোমরা প্রিপারেশন নাও, এবং সবাই খুব ভালো পরীক্ষা দাও।
স্যার, ক্লাস
অসংখ্য।
এক্সামের ডিউরেশনটা স্যার কতক্ষণ হবে স্যার?
এক্সামের ডিউরেশন 2 আওয়ার্স,
আচ্ছা,
অ্যারাউন্ড 2 আওয়ার্স।
20 নাম্বারে না স্যার, 20 মার্কস?
হুম হুম।
আচ্ছা
4 and 16।
জি স্যার।
অনেক ভালো থেকো। হ্যাঁ, থ্যাংক ইউ।
স্যার আমাদের কুইজের মার্কস একটু দিয়ে দিন স্যার পারলে, যখন
ঠিক আছে,
ইচ্ছা। ওকে
ঠিক
স্যার।
আছে, ঠিক আছে, থ্যাংক ইউ।